Alright, so one of the things that I said we would do today is to go over some of the metabolic tests. So that's still loading, but what's going to be up on the board are the same ones that I've got on these sheets here, and what I'm asking you to do is to work alone or with somebody else. I'm just going to give you that pile because I don't count. To work on identifying in these tests, what is it testing for? So in particular, what kind of metabolism? So like, is it going to help us to determine something about aerobic respiration or anaerobic respiration or fermentation or something like that? I'll get more. I'll come over that way. And we'll take about 15 minutes to work on this, and then we'll go over them together. So. What's that? Yep, those are yours. So you should be able to identify a positive and negative result, what it actually means, the interpretation of it, and then what's it actually testing for. All right, everybody have one? Okay. What? Okay, go. Betcha, otherwise you wouldn't be spending time on them. Of course, the other one that's not listed on here, but you're expected to know, is gram staining. Okay, so what's a gram positive, what's a gram negative, what's the morphology? It's all fair game. Yes, Jasmine? You should be familiar with the steps, but like timing and stuff, no, that's okay. But you should be familiar with like crystal violet, safranin, iodine, decolorizer, etc., etc. Okay. Okay. So let's start with these two over on your right, the lactose phenol red and the methyl red. Lactose phenol red tests for what? Lactose fermentation, and the key point here is that Jaden also mentioned what the carbohydrate is as well. Okay, so if it's glucose phenol red, it's testing for glucose fermentation, so on and so forth. In this image here, the far left is an uninoculated culture. Ours appear a little bit more red than this, and there's a reason for that. Our water is just a little bit more basic than what they're using in this experiment. And then also they show E. coli, Shigella sonii, and Pseudomonas aeruginosa. Okay, cool. I wanted to show one picture real quick though, just so you have an idea. What color does this turn when fermentation occurs, typically? The phenol red, sorry. Yellow, right? Okay, so let me pull up an image real quick. There, there we go. Okay. So here are phenol red tests. Obviously, it's British because, oh gosh, this side. They're using commas instead of periods as a decimal point. That's a British thing. Okay. So pH 8 down to 6.0. Typically with phenol red, we see a color change to yellow right around 6.7 or so. Typically, ours are starting out at about 7.3 or 7.4 when we use them in the lab. So that means that there's some sort of acid being produced, right? And that acid is an indicator that fermentation has occurred. Anywhere below a pH of 6.6, 6.5, 6.4 is going to be yellow, regardless of how acidic it is. In this case here, and you can tell better on your sheets, I think, than you can on this image, which ones have undergone some form of fermentation? E. coli and the Shigella sonii. Yeah. Which one has performed, which one do you know for certain has performed heterofermentation? Why do you know that? Okay, so what does heterofermentation mean again?
Two results.
Yeah, at least two products, if not more, being formed. And in that one, we know not only an acid is being produced, but gas is being produced as well. So we know E. coli is a heterofermenter of lactose. For Shigella, we can't really tell. We know at least it's only producing acid, but we don't know if the acids that are being produced are just a single acid or if it's 2 or 3 different types of acids. Pseudomonas, is it capable of fermenting lactose? No. Okay. Does it mean it's, and oftentimes, as you'll see on Wednesday, we perform at least a couple of different of these phenol red tests. So the ones we do in lab, glucose, sucrose, and lactose are the major 3 ones that we do to check for fermentation. So if you're trying to keep score of what this all means, type of metabolism, fermentation, lactose fermentation. Results in interpretation, yellow, not yellow, gas bubble, not gas bubble. What does that actually mean? Okay, and in terms of where the organism's a heterofermenter, potential heterofermenter, it ferments, it doesn't ferment. What does methyl red test for? Stable acids. In other words, at what point does methyl red change colors? What pH? Matt? 5. Oh, that was not a hand raise, that was a number. Gotcha. Perfect. 5. Below that, it's what color? So below 5, it's red, actually. It's red. Above 5, it's yellow. So this is one of the common areas where people will get confused between methyl red and phenol red, is that methyl red is red below a certain pH, whereas phenol red is red above a certain pH. So you just want to kind of keep that in mind and keep track of that. Oftentimes we don't perform a methyl red test until we've gotten a positive for glucose fermentation in the phenol red test. And in the methyl red test, as was mentioned, what we're testing for are whether the acids that are being produced are stable or not. So typically, ones that are stable are acetic acid, lactic acid. Media will stay red in those cases. If not, they get broken down, leading to more neutral products. Just to kind of explain this a slightly different way, I should not have closed that. Damn it. Hey, look, the exam's on there as a recently viewed thing. I wrote it over the weekend. It was so much fun. All right. All right, so here's an example of looking at phenol red of a organism that would be MR positive versus one that's MR negative. And again, oftentimes, not that you need to know this for the exam, but those that are MR negative are oftentimes VP positive or Vogts-Proskauer positive. This one's showing that below 5, the methyl red is a reddish color. Above 5, it's more yellow. But note, in both of these examples here, it's showing that regardless of whether those acids are stable or unstable, where does the pH stay? It's below 7, and so this is how an organism can be still positive for positive for phenol red fermentation, but negative for methyl red. As if it takes those unstable acids and converts them into something else, we can get that kind of result. Where methyl red is negative, but they're still positive for fermentation. Okay, so these obviously are both tests about fermentation. The methyl red tells us a little bit more about, think of it kind of like a dichotomous key. What type of acidic byproducts are produced during fermentation? Allie?
So for the first one, it's like testing the fermentation of lactose. For the second one, is there like multiple different acids?
So usually in methyl red, I should state that the carbon source in the methyl red test is glucose. So it's usually testing for if an organism ferments glucose, what are the potential breakdown products? And again, we kind of group them. The stable ones are like lactic acid. The unstable ones are short chain fatty acids that get broken down. But you don't need to know that. I would just say know it as, are the acids stable or are they not stable? Other questions on these two?
Right, so it's considered stable if it's a positive result.
Yes, exactly. Jasmine.
So you said it can show that it's positive for
Phenol red. Actually, so yeah, it can be positive both for phenol red, and positive for methyl red at the same time, or you can get where it's positive for phenol red, so fermentation has occurred, but methyl red negative. That the products that are produced during that fermentation are unstable and are broken down further. There's never a time, though, where you'll find methyl red positive and phenol red negative. Because the phenol red just detects, is there any acid at all produced? Yeah. Callie. Hint, hint, it's in the name.
Cytochrome oxidase.
Yay, it tests for oxidase, cytochrome oxidase. Positive result? Blue, purple. Negative result, no color change. This one, the test probably went a little too long. And I mention that only because this is one that John probably mentioned to you is time sensitive. It has to be read within about a minute or so. Not important for the exam. I will make sure that the positive or negative for this one is very clear. So test for cytochrome C oxidase. Where do we find that?
The
Electron transport chain. So that means that it's telling us about respiration. Which form? Yeah, primarily aerobic. So generally true, organisms that are positive for cytochrome C are obligated aerobes. They only perform aerobic respiration. That's a general truth. There are exceptions to that, though. So generally, organisms that are positive for oxidase only perform aerobic respiration. Okay, the indole test. What's this one test for? Yeah, so it's presence or absence of indole. The Kovacs reagent that we add, or the indole reagent, reacts with the indole. It's really, and another way to define that is it's testing for tryptophan breakdown, or the release of a couple of different, or production of a couple of different products from tryptophan. So in this particular test, what happens is tryptophan is in the media, tryptophan. And if an organism has tryptophanase, the enzyme, it breaks down tryptophan into indole, which is what we actually detect in this test. It also produces ammonia and pyruvate. What's going to happen to that pyruvate? If we wanted to produce energy with it, what has to happen to that pyruvate? It needs to be broken down. And where does pyruvate's last step of glycolysis, where does it go? Krebs cycle. So in other words, I bring this one up because this actually tells us something about the microorganism. One, it tells us, can it use tryptophan as a carbon source? But secondly, what are the only ways in which tryptophan can be metabolized to produce energy? If pyruvate's one of the products, which ways are going to yield to energy production here? So what pathways, how many ATP do we get in glycolysis? 2 per glucose, right? How many do we get in Krebs? 2 more per glucose. And then we get many more in respiration. If an organism breaks down tryptophan into indole, ammonia, and pyruvate, pyruvate's the last step of glycolysis. Therefore, if tryptophan's used as a carbon source, do we produce any energy from quote-unquote glycolysis? No. It's as if it's bypassed that whole 10 steps altogether. In other words, going to fermentation, really it has no net effect in terms of the amount of energy yield. So tryptophan, the only way that a cell can generate energy from tryptophan is breaking it down by respiration, either aerobic or anaerobic respiration. Because to be able to yield any energy from that pyruvate, it's got the carbons on it have to be fully oxidized. We get bunches of electron or reduced coenzymes NADH and FADH2, which will eventually provide energy in terms of oxidative phosphorylation. There are other carbon sources like this as well. Usually I think of it as if it's a carbon source that enters into those 3 pathways of glycolysis, Krebs cycle, or electron transport, if it enters after the energy yielding steps in glycolysis, the only way you can get further energy from it is through respiration. So for example, citrate, first molecule within the citric acid cycle. Only way to get energy from it, respiration. And this is true for fats, it's true for many amino acids, some nucleic acids, but not all of them. Yeah. All right, so those 4 tests and then gram staining are the ones that I'm expecting you to know. How will they show up on the exam? They're part of a dichotomous key. I've already developed the dichotomous key. You'll have to look at these results, be able to interpret them, and determine what the unknown organism is based on the dichotomous key and the sets of these types of tests. This particular worksheet should also help because you may be asked about what does this actually tell you about the organism, not just is it positive or negative, but what does that actually mean? Okay, so take a look at this over the next 48 hours. Danica. Yeah, so like if it were the oxidase test, that's a great example. If I were to say like what's the interpretation, has cytochrome C oxidase, and then what type of metabolism or what does this tell us about the organism's metabolism? Probably only does aerobic respiration. Yeah, exactly. Exactly. All right. Questions on these tests? We'll just start there real quick. Okay. Couple of announcements. 1, dichotomous keys due Wednesday in lab. Either hard copy to John or you can submit an electronic copy. There's a Dropbox on Blackboard under the lab tab. Only 1 per pair, so either you or your partner has to upload it into the Dropbox. Yeah, okay. 2, review tonight, 4:30 to 7, Sondrager 108. Bring food, bring a friend, bring a sleeping bag, whatever you prefer. Okay. I will tape it, so if you can't make it, I will tape it and upload it so that it's available. I hear there's an AMP one now at the same time. Who scheduled it? Oh, damn, Debbie. Okay. Don't tell her I said that. Let's see. 2 other things. 1, I mentioned the group surveys, or John did. If you want to have a say in what groups you're put in for this next round for Unit 2, let me know. I've already started kind of pairing groups up just based on what I've seen so far. Last thing, I forgot to mention this on Friday, and I'm sorry I did. Between now and, I don't know, 7 o'clock tonight, I'm going to make sure the lab's open. You or your partner, I need you to do 2 things. I have all of your plates of unknowns sitting out from 2-3 weeks ago. You need to do a new T-streak from that plate, and then you also needed to start an overnight culture. Okay, and I've got places for you to put them. So just go into the lab, take 10 minutes, start a culture, broth culture, and do a T-streak also, because you'll need those for Wednesday. I guess in that case then, if you guys don't get to, if you forget about it and don't do it until tomorrow, that's okay too. So, I'll have it open from the time we're out of here till about 7 o'clock tonight. And then, yeah, but don't leave right now. We still got 15 minutes. And then otherwise tomorrow, good times to do it, 8 to 10 in the morning, or sorry, 7 to 10. There's a lab from 8 to 10, but I've only got 10 people in there. And then probably from like 12 to 4 o'clock tomorrow as well. I'll make sure that it's open. Okay? All right. John will thank you. All right, let's do one more practice problem. Oh yeah, actually, real quick. Any last minute questions about the exam to address in class here?
Probably.
Okay.
Give me a second.
On Wednesday, when you guys come in, I'm going to have you sit in every other row, just so that I can get in between the rows to answer questions if they come up. So we'll do front row, third row, fifth row, seventh row, and then there's a row of seats all the way in the back too. So just every other row, so that I can sneak in between and answer questions. All right. Here is the question. 2 cultures of a facultative anaerobe, which means that it can grow in the presence or absence of oxygen, are grown in the same type of medium. One is exposed to air, the other is maintained under anaerobic conditions. Which of the 2 cultures will contain more cells at the end of the week? Why? I'll give you 5 minutes to work on this one. If you feel comfortable with it and if you can answer it, go back to the previous one. Or previous 2, sorry.